Now let's talk about the next question, which is question number 46. So this question says that you have to identify the correct pair representing the causative agent of typhoid fever from the confirmatory test for the typhoid. So basically, there are certain, agents has been given, causative agents has been given that causes diseases of particular, origin. So we have to just find out the confirmatory result, or you can say the confirmatory disease for them. All right, and that too we have to find it out for typhoid. Remember, we have to confirm it for typhoid. All right, now for typhoid, the test which is being performed, actually the causative agent for typhoid, let me tell you that the causative agent for typhoid, causative agent for typhoid. So the causative agent for typhoid is Salmonella typhi. So that is Salmonella typhi. All right, so this is actually a bacterium species that causes the typhoid disease. And the test which is widely used for categorizing, or you can say for testing the typhoid disease, the confirmatory test that is used for confirming the typhoid disease is basically the Vidal test. All right, that means this one. So you can say that the confirmatory test for typhoid, the confirmatory test for typhoid is Vidal test. And Vidal test is actually one such test that is based on the interaction between the antibody and the particular antigen. Antigen means that the particular bacterium. All right. So this is actually based on, you can say that this test is based on antigen antibody interaction. So on the basis of antigen antibody interaction, this is being confirmed. So here you can say that antigen are basically the bacterial species. Here the antigen which is being represented, these are the bacterial species. So antibodies will be produced in response to these bacterium species that will be binding up or detecting those bacterium species if it will be found in the body. All right. So out of all the options that you can see over here, so option C is the correct answer. That means Salmonella typhi is the causative agent and the test is Vidal test. All right. So that is the case. Now, question number 47. Question number 47 says that the Earth Summit that was held in the Rio de Janeiro, all right, in 1992, that was called for, all right, or that was called. So this test, basically this Earth Summit test, let me tell you that this Earth Summit has been basically conducted in Rio de Janeiro. For this is one of the direct questions that has been asked. So as such, there is no kind of explanations being required, but we can say that this summit has been taken into consideration for the sustainable development of resources. All right. So the main, you can say main focus of Earth Summit was basically the main focus of Earth Summit was to keep a check on the sustainable utilization of the resources. All right. And also to conserve those resources. So conservation, you can say, along with sustainable use of resources. Now, what do you mean by sustainable resources? Sustainable use. So this sustainable use means that uh, the resources should be used. We know that in our nature the resources are limited. So those resources like water or forest or soil or land, all those resources which are limited, that should be used in a manner which is actually going to be which that can be in a kind of a way that can be utilized for the present generation also, by the present generation also, and that can be utilized by the future generation as well, that can be preserved for the future generation as well. So the Earth Summit, the main target or the main focus of Earth Summit was this only. That means to conserve, and also along with to utilize, to sustainably utilize the resources. So out of all the options that you can see on your screen, so the first option says that for conservation of biodiversity and sustainable utilization of its benefit. Exactly. So this is the correct statement that has come out to be. The very first statement is the correct statement. To access threat posed to native species by invasive weed species, no, this is not the case. For immediate steps to discontinue use of chlorofluorocarbons, basically chlorofluorocarbon, the use of chlorofluorocarbon has been related with Montreal Protocol, but not with this Earth Summit. So definitely this is not the correct answer. And to reduce the carbon dioxide emissions and the global warming, again, this is also not the correct answer. So that's why option A is going to be the correct answer. Clear? Now, the next question is question number 48. Now, question number 48 says that the variations which are caused by mutation, as proposed by, Hugo de Vries, they are. All right. So, let me tell you that Hugo de Vries is one of the scientists, according to whom that he actually believed that that one of the reason for evolution is mutation only, or one of the reason for variations are mutations only. So you can say that according to de Vries, according to de Vries, mutations causes variations. So it's causes variations. So, according to him, mutation was one of the reasons that was responsible for causing variations in speciations, because he believed that and according to this concept only, he believed that mutations are, since these mutations they causes variations, that's why mutations are considered to be as a random process. That means a process that is very random, that can occur at any of the point of the time of the evolution, and that which is directionless also, that has no direction. That means that which organisms are going to be propagated, which organisms are not going to be propagated, that completely depends upon the extent of mutation actually. All right, so we can say that mutations causes variations, and mutations are random and directionless. Remember this. Directionless. They are having no directions. So basically, he believed this mutation is actually, since mutations causes variations, so variations ultimately lead to speciation only. So according to De Vries, mutation resulted into speciation also, and hence he called it as saltation theory. So that is also one of the main reasons you can say that De Vries believed that mutations causes speciation. That means formation of new species, and hence he called it as saltation. And hence he called it as saltation. That means, in his words, it was single step large mutation was there. Clear? This is the case. So out of all the statements that you can see, is it random and directionless? Yes. So important the very first important option is the correct one. That means A is the correct one. Then small and directional, no. Small and directionless, no. Random and directional, no. All right. So that's why the correct answer is option number A over there. Now, question number 49. Now, when we look into the question number 49, question number 49 says that which of the following can be used as a biocontrol agent in the treatment of the plant disease? So you have to look out for the biocontrol agent that is used for the treatment of the plant disease. All right. So there is chlorella, anabina, lactobacillus, and trichoderma. Now let me tell you, this chlorella is basically a unicellular alga. So this is generally being used as a food. So that is not used for a biocontrol agent. Anabina is used for nitrogen fixation. Again, that is also not used as for treatment of plant diseases. Lactobacillus is basically a lactic acid producing bacteria. All right, that has its own importance because it converts milk into curd. So the only option left here is the trichoderm. All right, so every species, that means all these species, if you just look into these three categories, that means chlorella, anabina, and lactobacillus, all these are economically beneficial species, you can say. Economical beneficial species. And if anything which is economically beneficial, then definitely it won't be causing any kind of diseases. All right. It won't be causing any kind of diseases. All right. So here only the last option is the trichoderma. So trichoderma, let me tell you that trichoderma is basically a fungal category. So, trichoderma is a fungus. So this is basically a fungus that is used as a biocontrol agent. Biocontrol agent developed for treatment of plant diseases. So this is the reason. And this is the question that has been asked from Microbes in Human Welfare chapter. And all these are economically beneficial species. Like as I said, chlorella is the unicellular alga, which is the richest source of protein. Anabina helps in nitrogen fixation. Then lactobacillus is a lactic acid bacteria. So all these are economically beneficial species. So they won't be causing any kind of plant diseases. All right, or they cannot be used as for treating for the treatment of the plant diseases actually. But trichoderma, since it's a fungus, so that is used as biocontrol agent for treating the plant diseases. All right. So this option, that means option D is the correct answer. Clear? Now, question number 50. So question number 50 says that which of the following immune responses is responsible for rejection of the kidney graft? All right. So which of the categories of the immune responses is actually responsible for rejection of the kidney graft? If this thing is being to be considered. Now, let me tell you that, our body has a very unique ability. All right. Our body has a very unique ability of differentiating or distinguishing the self cells from the non-self cells. Now self cells are those cells which belongs to the body itself, and non-self cells are those cells that doesn't belongs to the body. So for when any kind of organ transplantation is there, like kidney transplantation or any kind of organ transplantation experiment is being performed, so during that process, the cell mediated immune response, all right, that is actually responsible for any kind of grafting rejection. So if the grafting is successful, then it's fine. But if the immune response doesn't receives this grafting, that means doesn't accept the grafting process, so it is going to actually reject it. So it depends upon the category of cell, because all right, as I already told you, that our body has an ability of recognizing the self cells from the non-self cells. So on the basis of the particular kind of cells being grafted into the body, cell mediated kind of immune response, which is also called as CMI, that helps in the rejection of the kidney graft. All right. So we can say that the reason will be that our body is able to differentiate between self and non-self cells. All right. And so the cell mediated immune response is actually responsible for kidney graft rejection. Kidney graft rejection. All right. So what will be the correct answer? So the correct answer will be option number C over here. All right. Because humoral immune response, that is related with blood, inflammatory immune response, that is related with any kind of inflammatory reactions taking place in the body because of allergic reactions, autoimmune response are different. So that's why the option C is the correct answer. All right. Now question number 51. Now question number 51 says cells in G0 phase. All right. So let me tell you that we know that how the phases G0 is basically, let me tell you about the G0 phase first. So G0 phase is actually considered to be as question phase. This is known as question phase. All right. Or a phase in which that if there are some cells that does not want to enter the phase of cell cycle division, then then they directly just quit the cell cycle process, and they enter into this phase, like for example heart cells or neuron cells. These cells, they do not enter into cell division phase because they are already in a proliferated state. That's why. So if you just look into the options that has been given, so cells in G0 phase enters the cell cycle? No. Cells, whatever the cells that belongs to G0 phase, that never enters into the cell cycle, because if they will be entering into the cell cycle, then definitely, if they will be entering into the cell cycle, then definitely they will have to exit from it. They won't be able to exit from the process of cell division. That's why. All right. So this is the case. So they suspend from the cell cycle. Again, this won't be an option also. All right. And then terminate the cell cycle and then exit the cell cycle. So termination is also not a process. All right. So the last option left, which is exit the cell cycle. So as I already told you that G0 phase or a G0 phase is basically a question phase. All right. And cells, you can say that, and cells entering, or you can say cells of G0 phase are said to exit cell cycle. So they are known to exit cell cycle because they do not proliferate until they are told to do so. So these cells do not proliferate. Proliferate means to divide, or divide until called upon to do so. Like, for example, if I tell you, then there will be heart cells, then there are neuron cells, or neural cells, you can say. So neuron cells, these are the cells that enters, that are permanently that permanently resides themselves in the G0 phase only. They do not enter into the cell cycle process. All right, so option D is the correct answer. And question number 52. So question number 52 says that which of the following is a commercial blood cholesterol lowering agent? Now this is again a direct question that has been asked, and let me tell you that blood cholesterol lowering agent it has been asked about. So out of all the options like statin, then streptokinase, then lipases, and then cyclosporine A, statins are considered to be as the blood cholesterol lowering agent. All right. So this is one of the bioactive molecule. Actually, these statins are actually the kind of a bioactive molecules. It's a category of bioactive molecule actually, which is produced by a fungus, and that is of yeast category, which is called Monascus purpureus. So that's why it is used as a blood cholesterol lowering agent. So you can say that statins, which are a bioactive molecule, bioactive molecule is obtained from yeast, which is a category of fungus, and in yeast the category is Monascus purpureus. This is Monascus purpureus. So how does it lowers the this is basically used as a blood cholesterol lowering agent. So how does it do it? So these statins, they are basically responsible for competitively inhibiting the enzyme that is responsible for synthesizing the cholesterol. And once the enzyme will be inhibited, the cholesterol synthesis won't be there. All right. So you can say that, and is used as blood cholesterol lowering agent. All right. So blood cholesterol lowering agent. So you can say that what is the action, mode of action? So the mode of action is it acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. So once the cholesterol synthesis will be blocked, the blood cholesterol will be automatically lowered. So this is how they do it. All right. So that's why statin. Option A is the correct answer. Now, next question is question number 53. Now, question number 53 says that which of the following muscular disorders is inherited? Now, which of the following muscular disorder is inherited? This is also a category. You can say that out of the disorders that has been enlisted below, that means muscular dystrophy, myasthenia gravis, then botulism, and then tetany. Now, inherited disease this is talking about, or inherited disorder it is talking about. So this is basically the muscular dystrophy answer. The answer will be muscular dystrophy. And muscular dystrophy, as the word is suggesting, this is a disorder that is related to the degeneration of the skeletal muscles. All right. There is degeneracy in the skeletal muscles, and this is actually a why it is inherited. Let me tell you that it is inherited because it's a kind of a category of a genetic disorder only. And since this is a category of genetic disorder of skeletal muscles, that means degeneracy in the skeletal muscles, where this basically it results into progressive degeneration in the muscles. So you can say that muscular dystrophy, basically muscular dystrophy is a genetic disorder caused due to, you can say, prolonged, in fact, caused due to prolonged degeneration in skeletal muscles. Skeletal muscles. All right. So this is regarding the muscular dystrophy. Now let me tell you that what is this tetany or botulism, or you can say myasthenia gravis. Basically, myasthenia gravis is a kind of an autoimmune disorder. This is a category of autoimmune disorder. Autoimmune disorder, and this is actually not inherited. So this is an autoimmune disorder, myasthenia gravis, that results into, or that leads into, paralysis of the skeletal muscles, leading to paralysis of skeletal muscle, you can say. So leading to paralysis of skeletal muscle. All right. Leading to paralysis of skeletal muscles. While botulism, if I tell you, so it's a kind of a food poisoning disease, you can say. This is a category of food poisoning. So this is again won't be an option. So this is not an option. And this won't be also not an option because this is a category of food poisoning. All right. And tetany is a kind of a muscular spasm. This is a category of muscular spasm. So this is a muscular spasm. So this muscular spasm is actually caused because of low calcium in the body fluid. So since there is low calcium in the body fluid, so that ultimately results into tetany, cause of tetany. So this is the case, and the correct answer will be option number A, since this is a genetic disorder that is inherited. All right, now let's move on to next question, which is question number 54. Now, question number 54 says that you have to select the correct option with respect to the following sentences that has been given over here. All right. Now, uh, the first statement says that the 11th and 12th pairs of ribs are connected to the sternum with the help of hyaline cartilage. All right. So let me tell you that 11th and this is actually an incorrect statement. Why? Because 11th and 12th pairs of ribs are the floating ribs. All right. So they are not connected ventrally. That means they are not connected to the sternum. So that's why this is an incorrect statement. So option A is basically incorrect, because you can say that 11th and 12th pairs, 12th pairs of ribs are floating ribs actually. These are floating ribs. All right. So these are considered to be floating ribs, and so just they are not connected ventrally. All right, so that's why they are not connected ventrally or to sternum, you can say. So this is definitely an incorrect statement. Clear? Now, the second statement says that each rib is a thin flat bone or a flat thin bone, and all the ribs are connected dorsally to the thoracic segment or thoracic vertebrae, and ventrally to the sternum it is talking about. So basically, if we just look into the options or categorization, so the statement is saying that each rib is basically you can say it's a kind of a flat thin bone. Now let me tell you that all ribs have different different categories of ribs. They have different categories of connections, you can say. All right, like for example, if I just tell you about the vertebrosternal ribs. All right, so if there is a sternal ribs which are vertebrosternal ribs, so those are considered to be as true ribs, because they are dorsally attached to the thoracic vertebrae and ventrally attached to the sternum with the help of the hyaline cartilage. All right. And first pairs are actually considered to be, first of them, they are actually considered to be as the true ribs. So this statement and this statement, if you see like this one, that means option number C, that there are 7 pairs of vertebrosternal ribs and 3 pairs of vertebrochondral and 2 pairs of vertebral ribs. If we are going to give the explanation of this part, so we can say that next is All right. So basically, the reason will be that you can say that eighth, ninth, and 10th pairs of ribs do not articulate directly. They do not articulate directly with the sternum. Clear? But they actually join the seventh ribs. So, but you can say, but join the seventh ribs with the help of hyaline cartilage. Hyaline cartilage. Clear? This will be the case. So that's why these are considered to be as false ribs or vertebrochondral ribs. So these are false or vertebrochondral ribs. So this will be the case. All right. So again, this statement also is actually wrong. Now, the next statement says that there are 7 pairs of vertebrosternal, then 3 pairs of vertebrochondral, and 2 pairs of vertebral ribs. All right. Now, when it has been said that there are 7 pairs of vertebrosternal, then 3 pairs of vertebrochondral and 2 pairs of vertebral ribs. So we have already seen that the last 2 pairs, they are actually not connected ventrally to the, they cannot be considered to be as a ventral ribs because they are not connected ventrally to the sternum, and that's why they are considered to be as the floating ribs. All right. So this is basically this statement that says that 7 pairs of vertebrosternal, then 3 pairs of vertebrochondral and 2 pairs of vertebral ribs. This is actually the correct statement. All right, this is considered to be as the correct statement. That means that there is a presence of 7 pairs of vertebrosternal, which are connected ventrally. All right, first 7 pairs of the ribs, they are ventrally connected to the sternum. So that is true. Then 3 pairs of vertebrochondral are there. This is also true. And 2 pairs of vertebral ribs, which are ventrally not connected, which are considered to be floating ribs. So this is true. Now the last statement says that 8th, ninth, and 10th pairs of ribs, they articulate directly with the sternum. No, they do not articulate directly with the sternum, as the statement has been given over here, that they do not articulate directly with the sternum, but they actually join with the 7th ribs with the help of hyaline cartilage, and that's why they are considered to be as the vertebrochondral ribs over here. So, out of the 4th statement, option C is considered to be as the correct answer. All right. Now, the next question says that which of the following statement is considered to be as correct? All right. So, you have to find out the correct statement out of all these statements. Now, cornea basically consists of dense connective tissue of elastin and can repair itself. All right. Then the second statement says that cornea is convex, transparent layer which is highly vascularized. All right. Then the third statement says, you have to find out the correct statement, remember. Then the third says that cornea consists of dense matrix of collagen and is the most sensitive portion of the eye. All right. And the fourth says that cornea is external, transparent, and protective proteinaceous covering of the eyeball. Now, let me tell you one thing, that out of all these four statements, all right, see, cornea is basically that part of the eye which is considered, which is usually made of collagen and epithelium. All right. And this is actually considered to be as the main part, or you can say main sensitive portion of the eye. All right. So out of all these four statement, this third statement is considered to be as the correct one. So the statement will be that cornea consists of dense matrix of collagen and corneal epithelium. So this is usually made of collagen and corneal epithelium, which is the most sensitive part of eye, which is most sensitive part of eye. All right. So option C is the correct answer. Clear? Now, the next question is that this question says, question number 56. This says that you have to describe the characteristic. That means out of the following statements, describe the characteristics of the enzyme restriction endonuclease and identify the incorrect statement. So you have to basically identify the incorrect statement regarding all the statements regarding the restriction endonuclease enzyme. Now, the first statement says that the enzyme binds DNA at the specific sites and cuts only one of the two strands. All right. So this is the correct statement basically. Up till this is the correct one, that restriction endonuclease is one such enzyme that binds to DNA at specific sites. So this is actually true. This is true. But does it cuts only one of the two true or one of the two strands? So let us just see whether it's true or false. Before, let us take a look into the other statement as well. The other statement says that the option B says that the enzyme cuts the sugar phosphate backbone at specific sites on each strand. All right. Now, this is considered to be as a correct statement because the cleaving, where the restriction endonuclease cleaves the molecule, it cleaves by recognizing the sugar phosphate backbone. All right. So this is again a correct statement. Then the enzyme recognizes a specific palindromic sequence, palindromic nucleotide sequence in the DNA. This is also considered to be as the correct statement because one of the examples of the restriction endonuclease that has been given, that is regarding with Malayalam. All right. So palindromic sequence is such sequence which once you will read from left side or right side will remain the same. All right, so that is the case. So let me first tell you regarding some of the key points. See, restriction endonuclease is one such enzyme. Restriction endonuclease. Now, this is one such enzyme that cuts DNA at specific site or specific point, you can say. To specific site or specific point by recognizing a specific sequence. So by recognizing specific sequence. All right. Now, definitely each of the restriction endonuclease, they are unique because the restriction endonuclease actually inspects, the length of the DNA sequence. And once it is going to find its specific recognition sequence, it is going to bind to that DNA and cut two of the strands. That means the cutting will be there in both the strands. So that is how. So the next important point will be that restriction endonuclease functions by inspecting the length of DNA. So they actually function by inspecting the length of the DNA sequence and recognizes specific site, palindromic site, you can say, site or sequence in the DNA where it will be binding it, where it binds. Where this is going to bind. All right. Like then let me tell you the reason and then I'll just also tell you the example. So this is the case. Then it cuts actually the DNA on both strands. It cuts DNA on both strands. This is going to recognize that specific site, bind to it, and then cut the DNA on both the strands at specific point, and that too in the sugar phosphate backbone. So at specific points in the sugar phosphate backbone. All right, so this is the case. So like for example, if there is a sequence like this, all right, this is a sequence. So where the restriction enzyme will be actually acting, so the site of restriction enzyme is basically between, in this palindromic sequence, is basically between G and A. So it will be on both the strand. So this is the first strand and this is the second strand. So it will be on both the strand and the cleaving will be done on both the strand, not on the single strand. And that too by recognizing the sugar phosphate backbone like this. All right. So that's where the correct answer is option number, you know, sorry, this one, that means this if you will, you have to identify the incorrect statement. So the correct statements, this is a correct statement that yes, it cuts the sugar phosphate backbone. Then the enzyme recognizes a specific palindromic sequence, this is also a correct statement. And then the enzyme cuts DNA molecule at identified position within the DNA, that means specific site, restriction sites in the DNA, this is also true. And it cuts only one of the two strands that has been written over here, not only one of the two strands, both the strands of the DNA, it is going to cut it. That's why this statement is incorrect statement, and with regard to this, it will be the correct answer. Option number A is the incorrect statement. All right, so that will be the case. Now question number 57. Now, question number 57 says that phloem in the gymnosperms, they lack. All right, so phloem is basically a kind of a vascular tissue. It's a category of vascular tissue, or you can say it's a category of permanent tissue, you know, that is responsible for the translocation of food and minerals in the food and other dissolved organic molecules through phloem. So basically, phloem consists of sieve tubes, companion cells, phloem fibers, and phloem parenchyma. But when we are talking here about gymnosperms, so specifically, if we just see, then the gymnosperms, the phloem of the gymnosperms, they usually lack two specialized cells, and those are considered to be as sieve tubes. All right. So sieve tubes also and companion cells also. So basically, let me tell you that in angiosperms, if you are going to see, in angiosperms, if you consider, then phloem has sieve tubes, then companion cells, phloem fiber, phloem parenchyma. These are the constituent of phloem in case of the angiosperms. But when we talk about in gymnosperms, so in gymnosperms, if you will see, in gymnosperms, phloem consists of basically, or phloem has, in place of sieve tubes, there are sieve cells, but not tubes. So that's why they are unspecialized. So not tubes. All right. Then in place of companion cells, they have albuminous cells. So you can say that companion cells absent. Companion cells absent. But albuminous cells found. And apart from that, they will also be having the phloem fiber and the phloem parenchyma. So they also have phloem fiber and phloem parenchyma. Clear? So let us take a look that what are the compositions. So phloem and gymnosperms, they basically lack sieve tubes only, companion cells only, both sieve tubes and companion cells. So this will be the correct answer. Yes. Albuminous cells, they have the presence of albuminous cells and sieve cells. So since it is talking about here lacking. So remember. So that's why the correct answer will be option number C over here. All right. Now, question number 58. Now, question number 58 says that use of an artificial kidney during the hemodialysis may result in. All right. So, certain options are given regarding the hemodialysis process. So let us take a look into the different options that use of artificial kidney during the process of hemodialysis may result in. There is nitrogenous waste buildup in the body. The first option is nitrogenous waste buildup in the body. Second is non-elimination of excess potassium ions. Third is reduced absorption of calcium ions from gastrointestinal tract. Then reduced RBCs production. Now, which of the following options is considered to be as the most appropriate? So out of all the four options, you have to select the two appropriate options over here. Now, let me tell you that in case of hemodialysis, all right, where an artificial kidney is used for the process of dialysis, so this option which is saying that this one, that nitrogenous waste buildup in the body and non-elimination of excess of potassium ions, these statements are actually considered to be incorrect, because in dialysis there is elimination of urea and potassium from the body. All right. So this statement will actually be, both these statements, these two statements will actually be wrong. Why? Because during the process of dialysis, there is elimination of urea also, and then there is elimination of potassium also from the body. So you can say that A and B statements are incorrect because in hemodialysis, there is elimination of urea and potassium ions from body. So there is elimination of urea and potassium ions from the body. All right. While option, let us take a look into the option C and D. So option C says there is a reduced absorption of calcium ions from the gastrointestinal tract. So reduced absorption of calcium ions from the gastrointestinal tract. So yes, this will be a correct statement. This is a correct statement for the hemodialysis, because as soon as the calcium ions are also being eliminated, in this process of hemodialysis, actually calcium ions are also being eliminated during this process. So since calcium ions are also being eliminated, so there definitely will be a less absorption or a reduced absorption of calcium ions from the gastrointestinal tract, that means GIT part of the body. All right, so this is somewhere the correct statement. And the last is reduced RBC production, definitely, because there is reduced production of RBCs because there is reduction in the amount of erythropoietin hormone. And once the erythropoietin hormone is reduced, so definitely this is going to result into less production of RBCs also. So that's why this one and this one, that means option number C and D is considered to be as a correct answer. All right. So C and D are correct. So these are considered to be correct. All right. Now let's move on to the next question, which is question number 59. Now, question number 59 basically says that which of the following are statements are considered to be actually incorrect. All right, so which of the following statements are incorrect? Now the first statement says that viruses are obligate parasites. Now this is considered to be as an see, viruses are obligate parasite. If I just tell you, then basically these are something that are not considered to be as a parasite, because this is the constituency. See, if we just consider viruses to be as a parasite, and they are considered to be as a parasite because, and their hosts are very specific. All right, so they are very much concerned with their specific host. So viruses are actually considered to be as an obligate parasite because they have some specific host. So they possess specific host, you can say. So they possess specific host. And for this reason only, they are considered to be as an obligate parasite. So this is actually a correct statement. All right, this is a correct statement. The next statement is that ineffective constituent in viruses is the protein coat. So, infective constituent in the viruses is the protein coat. Now, let me tell you that if I just draw a figure, then viruses, they something look like this. So they have a body structure like this. Then this is something that an overview that I'm drawing like this. Then inside basically they have the crystal like structure, and inside it they basically have the presence of, let me tell you, the genetic material which is present inside it like this. And they have the presence of, tail fibers like this, tail pins, then tail fibers like this. All right, so this is the protein coat. So the protein coat is the non-infectious part of the virus. Actually, the genetic material which is present inside the protein coat, if I'll tell you. All right, so protein coat here, and here is basically the genetic material. So this is the RNA, you can say. All right. So this RNA is the infectious infectious agent, not the protein code. So this is actually the incorrect statement that has come out to be. This is the incorrect statement. All right. So RNA is the infective constituent in virus. Clear? Now, prions consist of abnormally folded proteins. Yes, this is also a correct statement. Yes, prions are actually prion is considered to be, prions are actually those uh, particles, or you can say those agents that causes infections through proteins. So that is the case, and here they lack, prions are actually the proteinaceous, they are also called as short they are also considered to be as proteinaceous infectious particle. That means they cause infection through their proteins but not through their genetic material, because they lack genetic material, they do not have any kind of genetic material. That's why. Then viroids, they lack protein coat. So this is also correct statement that yes, viroids also lack any kind of protein. That's why they are called. They are known to have naked RNA. Since they lack protein coat, so they have naked RNA. So this statement is also considered to be as correct. So the incorrect statement is basically option number B over here. All right, this one incorrect statement. So I have to find out the incorrect statement over here. Remember. Clear? Now, question number 60. Question number 60 says that due to increasing airborne allergens and pollutants, many people in the urban areas, they are suffering from respiratory disorder causing wheezing due to. All right. Now let me tell you that wheezing is basically a process of asthma. All right. And asthma is a result, or it is a process where there is difficulty, disorder, not process we can say, but this is a disorder that results into difficulty in the process of breathing, that ultimately causes wheezing due to inflammation in the bronchi and the bronchioles. So the very first important option is the correct answer that has been over there. That means inflammation of bronchi and bronchioles, because asthma is one such disorder, respiratory disorder, which is caused because of allergens. Remember, so this is here allergens, it is talking about here allergens, so this we have to keep in mind. All right, so you can say that asthma, just a second, asthma is a respiratory disorder resulting into difficulty in breathing, and that too due to wheezing. Wheezing or causing wheezing that results to inflammation, that means swelling of the bronchi and bronchioles. So inflammation of bronchi and bronchioles. All right. And this is primarily caused because of allergic reactions only, and many people they suffer from such kind of respiratory disorder. So that's why it is the option A, which is the correct answer. All right. Now, let's talk about the next question, which is question number 61. Now, this question says that which of the following sexually transmitted diseases, that means which is shortly termed as STDs, is not completely curable. So it is talking about the options, that means the options which is of the STD which is not completely curable. All right. Now, the given options are genital warts, then there is genital herpes, then chlamydiasis, and gonorrhea. All right. Now, let me tell you that out of all the four options that has been mentioned over here, now genital herpes is considered to be as the sexually transmitted disease which is not completely curative, or which is actually not curable. All right. Why? Because this is actually caused by the type 2 kind of herpes simplex virus, which is called as HSV. All right. So these genital warts are a kind of a curable disease, curable STD, because that is caused by human papilloma virus. Then chlamydiasis is caused by chlamydia, and gonorrhea is caused by Neisseria gonorrhea. So that is actually all these are curable. So we can say that genital herpes is caused by type 2 herpes simplex virus. So this is actually caused by type 2 herpes simplex virus, which is shortly termed as HSV. All right. So right now, if you're going to see, then this is actually this genital herpes. There is no cure as such available for genital herpes, but for genital warts, chlamydiasis, and gonorrhea, you know the vaccinations, then medicines are available for treating these diseases. All right. So this is one such case, and let me tell you that there are other diseases also. There are other categories of STDs also which are considered to be not curable, and it basically includes STDs like, you can say, STDs like hepatitis B and HIV. So hepatitis B and HIV, that means human immunodeficiency virus causing AIDS, is also not curable. This is also not considered to be as a curable disease. All right. Diseases. So that's why the correct answer here will be option number B. However, these genital warts, chlamydiasis, and gonorrhea, these are curable. Clear? Now, then let's move on to the next question. Now, question number 62 says that how does steroid hormone influences the cellular activities? All right. Now, if you now this is a question which has been asked from your endocrine glands chapter, and if you people just remember it, then there are two categories of hormones that we have come across. One is the proteinaceous hormone, and the other is the steroid hormone. All right, and both hormones have different kind of cellular activity that they possess actually. So here it is talking about the steroid hormone. Now let me tell you that steroid hormones are considered to be as the lipid soluble hormone, because they can cross the plasma membrane and they can just cross that barrier of the membrane. All right, so you can say that since these steroid hormones, so since steroid hormones are lipid soluble, because the plasma membrane is made of lipid only. So since they are lipid soluble, so they can directly enter into the cell. So they can directly enter into cell by crossing the plasma membrane, which is actually made of phospholipid bilayer. All right. Now, once it directly, since it can directly enter into the, cell inside this cell cytoplasm by crossing the plasma membrane, which is actually made of lipid molecules. So what they do is that once they enter into it, into the cell, they are going to bind with the, certain receptors which are present inside the cell, or you can say rather inside the nucleus. Those are actually considered to be as the intracellular receptors. All right, so after entering, basically after entering, they bind with intracellular receptors, because they need to be carried on towards the nucleus for affecting the cellular activity. So that's why they need to bind up with the intracellular receptors that are found in the nucleus. All right, and once it binds it there in the nucleus, they are going to form a hormone receptor complex. So they bind with the intracellular receptors in the nucleus and thus forms hormone receptor complex. Hormone receptor complex. And once this complex will be formed, this complex will further interact with the genome or the DNA of the cell, and in turn it will be forming, it will then be responsible for the process of transcription, through which it will form mRNA, and then it will form then via process of translation it will form that particular proteins. All right, so you can say that this hormone receptor complex, this hormone receptor complex further interacts with DNA and is responsible for transcription and translation. So they will further carry on the process of First, it will carry out the process of transcription, and then it will carry out the process of translating those effective proteins that are required for the cell. So this is how they do it. Now let us just take a look into the options. What are the options that has been given? So the first option says binding to the DNA and forming a gene hormone complex. So with this description that has been explained over here, this statement seems to be the correct statement, but let us take a look into the other statement as well. All right. So the second says activating cyclic AMP. Cyclic AMP is a kind of a secondary messenger, let me tell you, which is found on the cell membrane. But there is no requirement of secondary messengers over here because as the word is suggesting that steroid hormone, they can directly pass the cell membrane. They do not require any kind of membrane proteins to help them. So definitely this won't be an option. All right. Then using aquaporin channel as second messengers, again, this also won't be an option because they do not require any kind of channel proteins or carrier proteins. So that's why. Then changing the permeability of the cell membrane, not an option. All right. Because there is no need to change the permeability of the cell membrane because they can directly diffuse into it. All right. So with all the statements, statement 1, that means option number A is the correct answer. All right, now let's move ahead with the next question, which is question number 63. Now, question number 63 says that given below are the statements, regarding the annual rings in the trees, and you have to find out the false statement. That means not true. So, which of the following statements given below is not true about the formation of annual rings in the trees? Now, let me tell you, annual rings basically are those rings in the trees that estimates the age of a tree, that are also considered to be as growth rings, or that is also known as to be an estimation. In order to estimate the age of a tree, annual rings are counted. So basically, annual rings also called as growth rings gives an estimate so it basically gives an estimate of age of tree. All right. An annual ring is actually a combination of spring wood and autumn wood, you can say. So this is actually, annual rings are actually a combination of, it's a combination of spring wood alternating with autumn wood. All right, so this is the case that you need to know it. Now, let us take a look into the other information. Now, so this says that differential activity of cambium causes light and dark bands of the tissues early and late wood respectively. So this is actually a correct statement because growth rings are actually formed as a result of the differentiation activity or the seasonal activity of the cambium. So this is actually a correct statement that, growth rings or annual rings, growth rings are formed as a result of differentiation activity of cambium. Differentiation activity of cambium. This is actually a true statement. All right. So this is a correct statement. Now the next statement says that activity of cambium depends upon the variations in climate. Definitely, the activity of variation definitely depends upon the variation in the climate, because if the climate will be warmer or hot, then the activity of cambium will be very fast. And if the climate is cooler, or you can say cold conditions are there, then the activity of cambium will be slow, and hence the division of cambium will be less. So definitely the cambium activity you can say is a you can say that this is a true statement. So we can say that cambium activity, just a second. So cambium activity depends on climate. Depends on climate. Like in spring season, it is more active. So, for example, if I say, then in spring season, cambium is very active. And it basically forms more number of secondary xylem, forming more number of secondary xylem. All right. But in autumn season, if I tell you, then in case of autumn season, the cambium is less active. So cambium is less active, and therefore less number of secondary xylem will be formed. So definitely this is also a true statement. Now the next is annual rings are not prominent in the trees of temperate region. Now this is actually considered to be as an incorrect statement, because annual rings are formed in all types of trees, whether it's a temperate tree or whether it's a tropical tree, all regions. It just depends upon the climatic condition. Like in tropical region, the annual rings will be more, but in case of temperate region, the annual rings will be less, but yes, they are prominent. So definitely this is the incorrect statement that we can see actually, the incorrect statement here. So you can say that now the next statement says, let us take a look into the next statement also, which is option number D. Annual ring is a combination of spring wood and autumn wood produced in a year. So definitely, as you can see over here, I have already written this, statement over here, where you can see that annual ring is a combination of spring wood and autumn wood that is actually found in a year. So this is also a correct statement. All right. So we can say that option C is considered to be as the incorrect statement. All right. And for the reason that we can give for that is, in, or rather you can say that just a second. Yeah. So you can say that annual rings are prominent are prominent in trees of temperate region. Temperate region. However, the prominency is less, you can say. Why? Because the weather conditions are little cooler. That's why. However, the prominency is less. Or number of annual rings, you can say, is less. in these trees are less. All right. So this is the reason. So the incorrect statement will be option number C over here. Clear? Now let's move on to the next statement, which is next question, which is question number 64. Now question number 64 says that what is the direction of movement of sugars in the phloem? All right. So basically, phloem is one such vascular tissue. This is a permanent tissue that is responsible for the translocation of sugars in the trees. So phloem, which is a category of a permanent tissue, complex permanent tissue. So complex permanent tissue that helps in translocation of sugars and other food as well. All right. Now regarding the movement, if I tell you, so the movement in case of phloem, the movement of sugars in case of the phloem is considered to be not upward, all right, no downward, not it is not multi-directional, but it is considered to be as a bidirectional transportation. Why? Because the movement of these sugar in the plant body, it actually depends upon the source and sink relationship. All right. So it depends that in some plants, the source will be the leaves, the stems, and the sink will be the roots. But in some plants, where modified stem is being found, where modified roots are being found, so there in in such categories of plants, the roots basically store food. So there the roots act as source, and the sink is a different part of the plant body. So via this way, so this basically depends upon the kind of a source and sink relationship that is actually variable in the plant body. So you can say that the direction of movement of sugars in phloem is bidirectional. Why? As it depends on the source and sink relationship. Now, what do you mean by source and sink? Source is basically that part of the plant body where food is synthesized, while like leaves or green stems. And sink is considered to be as that part of the plant body where the food will be ultimately stored. So it actually depends upon the source and sink relationship, which varies in the plant body. Which varies in a plant body. All right. So the option will be option number C over here. Now, next question is question number 65. Now, question number 65 says that the shorter and the longer arms of a submetacentric chromosome are referred to as. Now, let us first draw the submetacentric chromosome. So, basically, submetacentric chromosome is that chromosome where the centromere is slightly shifted from the center like this. All right, or we can say, we can like draw the centromere here first, and this is going to be like this. All right, so this is an example of a submetacentric chromosome where a submetacentric chromosome so here centromere is slightly shifted from the center, or slightly shifted to one side, you can say. Center of chromosome. All right, so this is the submetacentric chromosome. Now, if I tell you the shorter and the longer arm, so this one is considered to be as the short arm, and this one, of course, is considered to be as the long arm. So this is the short arm, and this is the long arm. Clear? Now, the short arm in case of the submetacentric chromosome is called as P arm. This is called as P arm, while the long arm is called as Q arm. P arm is actually, P basically stands for, this petite term. So P arm and then Q arm. All right. So let us take a look into the options that has been given. So if you can see, so the shorter and the longer arms of the submetacentric chromosome, they are referred to as P and Q arm respectively. It's true. The first statement, the first option is the true one. So then Q and P arm, no. M and N arm, not at all. S and I arm, no. All right. Now let me tell you that this submetacentric chromosome and is basically formed as a result of the shifting of the centromere slightly towards, it is shifted to one side from the center of the chromosome, and because of that only the upper arm becomes short and the lower arm becomes long over here. So it's a category, this submetacentric chromosome is actually a category of heterobrachial chromosome. This is a heterobrachial chromosome. That means basically two types of arms are found. Chromosome. All right. So this is the case. Now, next is question number 66. Now, question number 66 says that which of the following methods is the most suitable for disposal of nuclear waste? Now, most suitable for disposal of nuclear waste, it is talking about. Now, let me tell you that we all know that nuclear waste or are a category of radioactive waste. So this nuclear waste, the composition of nuclear waste is basically the radioactive material or the radioactive waste. All right. So definitely these waste, out of all the waste that we come across, these nuclear waste, they are the most hazardous of all the waste. So definitely these are, if you just look into the statements that has been given over here, so it should be bury the waste under the Antarctic ice cover, then dump the waste within rocks under the deep ocean, then bury the waste within rock deeps below the earth's surface, shoot the waste into space. Now, out of all the convenient option, if you're going to see, then Antarctic ice cover and under deep ocean, these two options are practically impossible because this cannot be carried out. Because if this will be done, that means disposal of the nuclear waste will be done under the deep oceans, then this is definitely going to harm all the aquatic organisms and plants and marine animals and plants. So definitely the first two options are not a very good option. All right. Then the option D says shoot the waste into the space. Now this also again is a kind of not a very well-to-do option, because shooting the waste into the space won't give a true solution for the disposal of the nuclear waste. And the only option left is bury the waste within the rocks deep below the earth's surface. So this is going to be the most suitable, you can say, most suitable method for disposal of the nuclear waste, which is a process in which these nuclear waste, they are usually being shielded inside the containers. All right, so you can say that nuclear waste are a category of radioactive waste. So since these are a category of radioactive waste which are very hazardous, which are very hazardous, so you can say, so these wastes are actually stored in suitable shielded containers. So these wastes are stored or packed in suitable shielded containers. Shielded containers, and then buried inside the earth surface, and then deeply buried inside the earth's surface. And it should be approximately 500 meters deep. All right, then only that won't be able to harm the organisms. So that's why the correct option will be option number C over here. Now next question is question number 67. Now question number 67 says that you have to match the column with the match the column kind of a question has been given, and column 1 to be matched up with column 2. Now in this question, this is basically a question of the ECG that has been asked over here. All right, so let me tell you about the ECG first. So ECG basically stands for the electrocardiogram that measures the it's a electrocardiogram is basically a graphical representation that gives the heartbeat movement basically. All right, and the machine that measures it is called as the electrocardiograph. And there are three waves that are found in the ECG: P wave, QRS wave, and then T wave. Now let us take a look that what does this ECG basically tells. Now the first is basically the P wave. All right, and if you just look into the option, so in the ECG machine, let me tell you that in ECG, which is electrocardiogram, P wave usually represents the depolarization of the atrium. So P wave represents depolarization of atrium. All right. Now, depolarization of atrium basically means the contractions that will happen in the atrium because of the atrial systole, or the sinoatrial because of the sinoatrial node. All right. So this basically represents this one. So P wave will be representing the depolarization of atrium over here. All right. Now the QRS complex is the main important complex, because by counting the QRS complex only, the number of heartbeat is calculated by counting the number of the QRS wave in the graph. So that is the case. So you can say that QRS complex, and this QRS complex, this is going to represent the depolarization of the ventricles. All right, so that means contractions that will happen in the ventricles. So this will be correlatable to this one. So QRS complex represents depolarization of ventricles. All right. And the last wave, which is T wave, basically, how the graph is being drawn. I'll just draw a figure over here so that you can have an idea. This goes like this. So this is something like this. So this is basically the P wave, then this is Q, R, S, and this becomes the T wave over here. All right, so this is the ECG, this is the graphical representation that I have drawn so that people can have an idea. All right, so this P wave represents the depolarization, QRS represents the depolarization of ventricle. Yeah, now T wave is going to represent the coming back of the ventricles into its normal state, basically. That means from the excited to normal state. So the opposite of the depolarization will be repolarization of the ventricles. So this will be option C over here. So you can say that T wave represents repolarization of ventricles. That means the ventricles now will be in a relaxed state. Again, getting ready for starting the process of cardiac cycle. So repolarization of ventricle, that is coming to normal state, from excited state. So coming back to the normal state. And the last option says the reduction in the size of the T wave. Now, reduction in the size of the, if there will be a reduction in the size of the T wave, then this will indicate, a kind of a defect, because any deviation in this entire ECG from the normal, process of waves will indicate a kind of, disease in them. So definitely this says that the reduction in the size of the T wave is an indicator of coronary ischemia. And coronary ischemia is basically it's a kind of a disorder in which there is insufficient supply of oxygen in the body. All right, so you can say that reduction in size of T wave indicates defect or disorder. Disorder related to heart. And so it is relatable to coronary ischemia. All right, which is insufficient supply of oxygen. All right, so this is going to match up with the option over here. Clear? Now let us take a look into the further options. So P is going to match up with A. Sorry, basically A is going to match up with fourth option. So A is going to match up with 4. So here and here are there. Then B is going to match up with first. B is going to match up with first. Then here, here, and here. Then then C is going to match up with second option. C is going to match up with this option. And then D is going to match up with third option. So D is going to match up with third option. So out of all the 4 options, this one is the correct answer that you can see. All right. Now, let's move on to the next question, which is question number 68. Now, question number 68 says that which of the following statements regarding the post-fertilization development in the flowering plants is incorrect? Now, let me tell you, post-fertilization development means the development which will happen after the process of fertilization has taken place. That means post-fertilization products are formed in case of post-fertilization developmental process in flowering plants. So that is incorrect. So zygote develops into embryo. So this is a correct statement. All right. So let me tell you that uh, changes during post fertilization. What are the changes during the post fertilization? If I tell you, then it will be very simple that ovule will be developing into seed after fertilization. So this develops into seed, then ovary develops into fruits. Ovary is going to develop into fruits. Then zygote, of course, zygote, which is the process of zygote, which is the product of syngamy of the double fertilization in flowering plants. That zygote develops into embryo, which is diploid. Clear? Then the central cell that has been written over here, as you can see, central cell is basically the secondary polar nucleus. So that undergoes the process of triple fusion. So this central cell that is going to develop into endosperm, which is the nutritive tissue found in the seed. And this is triploid. All right. Now let us take a look that what are the options that has been given. So zygote developing into embryo, this is correct. Central cell developing into endosperm, this is also correct. Ovules developing into embryo sac, no, not at all. This won't be an option because the embryo sac is the female gametophyte here over here, and this is the this is basically the pre-fertilization structure that which is formed in case of the ovule. So ovule develops into seed, not into embryo sac. All right, so this actually represents the female gametophyte. Remember. And then ovary develops into fruit. This is also correct. So you have to identify the incorrect statement. So incorrect statement, if you will see, then the incorrect of just a second. Yeah. So the incorrect of all the statements that we will be seeing it. So this is the incorrect statement. All right. That means the correct answer will be this one only. This statement only. Incorrect statement only. Because we have to find out the incorrect statement. That means option number C over here. C. This one. Now, next is question number 69. So question number 69 says that you have to select the correctly written scientific name of mango, which was first described by Carolus Linnaeus. Now, let me tell you that according to the rules of the binomial nomenclature that has been proposed by Carolus Linnaeus. So basically, Carolus Linnaeus proposed binomial nomenclature. And according to this nomenclature, and you can say that according to this nomenclature, mango is basically written as, the scientific name of mango, let me tell you, is Mangifera indica. Mangifera indica, where the Mangifera indicates the genus And Mangifera indicates the genus and indica indicates the species. All right, indica is going to indicate the species, and you have to note this point that M should be capital in the genus. That means the first letter of the genus should be capital, and the first letter of the species should be small over here. Clear? So this has to be the case. And apart from that, the person who has discovered it, that should also be mentioned. Like if this mango has been discovered by Carolus Linnaeus, so we are going to represent it as Lin over here, like this. All right, now let us take a look what is the correct one. So this is Mangifera indica Lin. Okay, so the first option has turned out to be the correct one. Mangifera indica, no, this cannot be true because here the scientist name is lacking. Mangifera indica, this again cannot be true because here I is capital over here. So this is a wrong option. And Mangifera indica Caroline, again, this is also wrong. Clear? So option number A is the correct answer. Now, question number 70. Question number 70 says that what is the fate of the male gametes discharged in the synergid? All right. So what is the fate of the male gamete that is going to discharge with the synergid? Now let me tell you that during the process of double fertilization, if we say, so during double fertilization in angiosperms, that means in flowering plants. All right. Pollen tube carries the two male gamete. We know this fact. It carries two male gametes. Now, out of these two male gametes, clear, if we just describe it, then these two male gametes are discharged into, all these two male gametes, they are discharged into synergids. So both male gametes, you can say, both male gametes are discharged into synergid. Clear? Synergids of the embryo cell. Now, out of these two male gametes, the first male gamete, this fuses with egg cell. So this is going to fuse with the egg cell to form a diploid product which is called zygote. All right. And this process is called syngamy. So you can say process known as syngamy. All right. While if I just talk about the second male gamete, so second male gamete is basically going to fuse with secondary nucleus, which is also called as central cell or polar nucleus. And since this is already diploid, so they are going to fuse with that, and the resultant product will be, they will be forming primary endosperm nucleus, which is shortly termed as SPEN, and this is triploid. All right. And this process, this entire process is known as triple fusion. All right, now let us take a look where the fusion has taken place. So what is the fate of the male gamete that is discharged into synergids? All fuses with the egg? No. So we have seen over here. One fuses with the egg and the other fuses with the synergid nucleus. Again, this is also a big no over here. One fuses with egg and the other fuses with the central cell nucleus. Again, this is true. We have seen over here, you can see one fuses with the egg cell and the other fuses with the secondary nucleus, which is also considered to be as central cell or polar nuclei. Clear? So this is the true statement. One fuses with egg and the other degenerates into synergid. Again, this is also a big no. All right. So this is the case. So the answer is option number C over here. So let's talk about the next question, which is question number 71. All right. So this question says that which of the following features of the genetic code does allow the bacteria to produce human insulin by the process of recombinant DNA technology? All right. So we have studied it also, and you people might have gone through this chapter also, where the human insulin is being produced through the process of recombinant DNA technology. And genetic code is basically one such code that has some unique properties or some unique features. So if we just look into the different options, the first option says that genetic code is redundant. Then genetic code is nearly universal. Genetic code is specific, and genetic code is not ambiguous. All right. Now, for a bacteria to allow the process of recombinant DNA technology, or to undergo the process of this recombinant DNA technology for producing human insulin, the very first and foremost important thing that is required in a genetic code is not to overlap. All right. So the important feature of genetic code is that it doesn't overlaps. All right. So it basically means that each nucleotide is a part of only one codon, or you can say that a single codon or a single nucleotide cannot be a part of two adjacent codons. All right. That means each codon codes for a specific amino acid, you can say. So this indicates what? This basically indicates the universal character of the genetic code. So for the process of producing human insulin, you can say that means for producing this human insulin, it is very, very important that the genetic code has to be nearly universal. That means if it is nearly universal, then a single nucleotide is encoded by only one codon and not by any other specific codon. All right. So that's why we can say that in recombinant DNA technology, DNA technology, all right, the bacteria is able to produce human insulin because the genetic code is nearly universal. So this indicates what? So this basically indicates that a single nucleotide cannot be a part of two adjacent codons. That means each nucleotide is a part of only one codon. It cannot be in a form of overlapping position. So adjacent codons. Clear? So with this, the correct answer is supposed to be the option number B over here. All right. Now, the next question is question number 72. Now, question number 72 says that which of the following pair of organelles does not contain DNA? All right. So let me tell you that organelles also contains the genetic material, and that is DNA. And if you just look into the different options like chloroplast and vacuole, then lysosome and vacuoles, nuclear envelope and mitochondria and lysosome. Now, let me tell you that out of all these options that has been given over here, mitochondria and chloroplast. All right. Mitochondria and chloroplast. These two are the cell organelles that have their own DNA and ribosome. All right. Have their own DNA. So they have their own DNA and ribosome. And that's why they are considered to be as the semi-autonomous organelles. So that's why called as semi-autonomous organelles. That means they autonomous. Yeah. So this means that they won't have to depend on the nucleus for encoding their DNA or the genetic material. So they are called as semi-autonomous organelles. Only these two organelles have their DNA, remember, not any other organelles. All right. So if you just look into the options that has been given over here, so it is talking about doesn't contains DNA. All right. So here the chloroplast contains DNA, but the vacuole doesn't contains DNA. All right. Then lysosome and vacuole both doesn't contains DNA. All right. Both of them doesn't contains DNA. Then nuclear envelope doesn't contains DNA. Mitochondria contains DNA. And here mitochondria contains DNA and lysosome doesn't contains DNA. So out of all the options that you can see, the lysosome and vacuoles, lysosomes and vacuoles does not contain DNA. Does not contain DNA. So these are the cell organelle that lacks DNA. All right. So that's why the correct answer here will be option number B. All right. Now, next question is question number 73. Now, question number 73 says that in some plants, the female gamete develops into ovary without fertilization. So this phenomenon is referred to as. Now, this is a very definitive kind of a question that has been asked. So this is a direct question, we can say. So basically, the phenomenon in which the female gamete will be directly developing into embryo without fertilization. So that process is considered to be as parthenogenesis. All right. Because parthenocarpy is a process in which, if I tell you, then parthenocarpy is one such process in which fruits are formed without the fertilization of the ovary. All right. So that is the case. Then syngamy is the fusion between male and female gametes. So this is also a sexual product. And autogamy is one such process in which there will be the fusion between the male and the female gametes that are identical to one another. All right. So out of all these options, parthenogenesis is the correct answer, because here only the female gamete undergoes the process of undergoes the development to form the embryo without the male gamete. That means without the actual process of fertilization. So you can say that parthenogenesis is a process in which So this is a process in which the female gamete undergoes development, and that too without fertilization. Without fertilization, it undergoes development without the fertilization with male gamete to form the product embryo. All right, so this is the case. And one of the classical examples of parthenogenesis, let me tell you, one of the classical example of parthenogenesis in which the female gamete only undergoes development, you know, it is going to undergo development without the fertilization will male gamete to form the product embryo, is the bees. Bees is a classical example of parthenogenesis. All right, now let's move on to the next question, which is question number 74. Now, question number 74 basically says that you have to consider the following statement. And that says that, just a second. Yeah. So the first statement says that coenzyme or metal ion that is tightly bound to the enzyme protein is called as the prosthetic group. All right. And the second statement says that a complete catalytic active enzyme with its bound prosthetic group is called as apoenzyme. All right. So you have to select the correct option, that out of which statements, whether both the statement are true or both the statement are false, or one statement is true or one statement is false. All right. Now, let me tell you that prosthetic groups are basically, let me tell you regarding the first statement first. All right. So the coenzymes or the metal ions and the prosthetic groups that is actually bound to the enzyme protein, that means the apoenzyme, that is considered to be as the cofactor. All right. And one thing to note regarding here is that now prosthetic group is one such group which is tightly bound to the enzyme. All right. So that is tightly bound to the enzyme, whereas the metals ions and coenzymes are those considered to be as those categories. Let me tell you that prosthetic groups are those groups that are tightly bound to enzyme. So these are tightly bound to enzyme. Remember this. These are tightly bound to enzyme. All right. While metal ions and coenzymes. While on the other hand, the metal ion and the coenzymes, they are transiently bound. Transiently means loosely bound. So transiently or loosely bound to enzyme. All right. And what is apoenzyme? Let me tell you. Basically, apoenzyme is basically the proteinaceous component, all right, of the entire enzyme. So let me tell you that this statement, all right, so if I talk about holoenzyme is the entire active enzyme, you can say. This is the active enzyme, which is holoenzyme, active enzyme. And this is a combination of apoenzyme plus cofactor. Clear? Now this cofactor, this is the protein component of the enzyme. This is the protein component, while the cofactor here can either be, this cofactor can either be coenzyme, can either be coenzyme. Clear? Or they can either be metal ions, or they can either be the prosthetic groups, or prosthetic groups that are bound to apoenzyme, that are bound to apoenzyme. That means the protein component of the enzyme. Clear? So this is the case. Now let us see that which statement is correct and which is false. Now here it is saying that coenzyme or metal ion that is tightly bound to the enzyme protein is called as prosthetic group. So again, this statement is considered to be as a false statement, all right, because if these if you just consider the prosthetic groups, then metal ions and coenzymes, they are not tightly bound, they are actually transiently bound. You can see over here, they are transient. Metal ions and coenzymes, they are considered to be transiently or loosely bound to the enzyme. So that's why this statement is false. And the second statement says that a complete catalytic active enzyme. So complete catalytic active enzyme is this one, holoenzyme. This is complete active enzyme. You can see over here. All right. So this is bound, this with its bound prosthetic group is called as apoenzyme. No. Apoenzyme is the enzyme, it is the only protein component of the enzyme, without which there will be no activity in the holoenzyme. All right. So definitely this statement is also false. All right. So both the statements are considered to be false. So if you just look into the options, both A and B are false. That means this is the correct answer. Option B. Clear? Now, let's move on to the next question, which is question number 75. Now, question number 75 says that the conversion of glucose to glucose 6-phosphate. All right. So the process of conversion of glucose to glucose 6-phosphate, the first irreversible reaction of glycolysis is catalyzed by. Now, let me tell you that glycolysis is a process, all right, in which glucose is partially oxidized to 2 moles of pyruvic acid. Partially oxidized to 2 molecules of pyruvic acid. Clear? So this is the definition. Now, what is the first? So now, since this process is talking about the first irreversible reaction, now let me tell you the first irreversible reaction is glucose getting converted into glucose 6-phosphate, which is this one. Clear? Now, the reaction or the enzyme rather that converts or catalyzes this process is called as the hexokinase enzyme. This is the very first important enzyme that has been mentioned in the book also, NCERT also. So it is the hexokinase that, that catalyzes this entire process. So that's why the correct answer here will be hexokinase. All right. Now, next question is question number 76. Now, question number 76 says that which of the following ecological pyramid is generally inverted? All right. Now, it is talking about the ecological pyramids, which is generally inverted, if I tell you. So, let me tell you the very first option that has been given, which is pyramid of energy. Now, pyramid of energy, we all have seen that pyramid of energy is never inverted. It is always upright. So, pyramid of energy is always upright, irrespective of any of the ecosystem. Remember, irrespective of any of the ecosystem, whether it's a forest or a sea or a grassland or anything. So that's why this is always upright. Clear? Now, the option B says pyramid of biomass in a forest. Now, pyramid of biomass in a forest is basically considered to be as an upright pyramid, because when you consider the pyramid of biomass in a forest, pyramid of biomass in a forest, all right. So here, the base is being formed by trees, a single tree which has a very huge mass. Then on tree, various kinds of fruit eating birds, they resided. So fruit eating birds are present, and on birds are present the parasites. Parasites are being found. So if you're going to look into the biomass, so tree has the greatest biomass, so that's why it's upright when you consider a single tree, because tree is the dominant vegetation in forest, so that's why this pyramid is also upright. All right. Now, apart from that, option number C, let us take a look. Option C says pyramid of biomass in a sea. Now, let us take a look. Now, pyramid of biomass in a sea, if you're going to do it, now this is going to be definitely inverted. Why? Because let me tell you that in case of the sea, the dominant vegetation, that means are the phytoplanktons, basically. So they are the phytoplanktons. Phytoplanktons is being succeeded by zooplanktons, like this. So they are having greater weight. Phytoplanktons are basically freely floating alga, so they are having lesser weight. Then there is zooplanktons. All right. Then zooplanktons are eaten by small fishes. Then there is small fishes which have bigger weight than the zooplankton. And small fishes are being eaten by big fish or big fishes. So that's why this is considered to be as an inverted pyramid. Clear? Like this. All right. So definitely this is the correct answer, and we have found our correct answer. And that's pyramid of number in grassland. Again, pyramid of number in grassland, if you will be considering on the basis of number it is talking about, so definitely the number of grasses will be high. So this is also considered to be as an upright pyramid. This is also upright. All right. So that's why the correct answer is option C. Now, question number 77. Now, question number 77 says that you have to consider the following statements and following features rather. So the first is organ system level of organization, bilateral symmetry, true coelomates with segmentation of the body. Now you have to select the correct option of the animal groups which possesses all the above characteristics. All right. So let us just look into the features now. Okay. Now you have to consider the following features. Now, regarding this, if we just say that organ system level of organization, if you are going to look into, so this is a higher category of organization. All right. So organ system level of organizations are seen in case of Annelida also, in case of Arthropoda also, and Mollusca also. But if we will be seeing the important features, all right. So the important feature is this, that if you just look into Annelida, Annelida shows the basic property of segmentation. All right, that means metameric segmentation. So if you just look into the segmentation, then true segmentation is found in, now segmentation is found in Annelida also, in Arthropoda also, and Chordata also. So in Annelida, Arthropoda, and Chordata. This is regarding the segmentation I'm talking about. And this organism actually, if we just categorize Annelida, Arthropoda, and Chordata, all out of all the options. Now the regarding the organ system level of organization, and they will be true coelomates also. They are also the true coelomates because they possess the true coelom category. All right, so that will be the case. So you can say that they also have, they also have organ system level of organization, then bilateral symmetry. They are all having bilateral symmetry. So they have bilateral symmetry in which the body will be divided into equal halves passing through a central plane only. All right, and then they are also true coelomes or true coelomate category. That means they have a well defined body cavity. All right, so if you just look into the different options, there is Annelida, Arthropoda, and Mollusca. Definitely this won't be the correct answer. All right, because segmentation is not found in case of Mollusca, so that's why it is not a correct option. Then there is Arthropoda, Mollusca, and Chordata. Arthropoda, Mollusca, and Chordata, again here this is not the option, and here Annelida, Mollusca, and Chordata, again this is not the option. So Annelida, Arthropoda, and Mollusca, Chordata, so this is the correct answer. So option D is considered to be as the right answer. All right, now question number 78. Now question number 78 says that which is the genetic disorder, or what is the genetic disorder in which an individual has an overall masculine development which is called gynecomastia and is considered to be as sterile. All right, now this is a question that has been asked from the genetic disorders category, and regarding the chromosomal disorders specifically, if we just look into the categorization. Now let me tell you that out of all the options that has been given, Klinefelter, then Edwards syndrome, then Down syndrome, and then Turner syndrome, basically, out of all the options, so individuals who have masculine development, that is called gynecomastia, that individual is being found if that individual is suffering from Klinefelter syndrome. Why? Because Klinefelter syndrome has a defect in their chromosome, and that too, that defects is being caused because of trisomic condition in the chromosome. All right, so if I just explain this part, that individuals with Klinefelter syndrome has an overall masculine development. So they have an overall masculine development and are sterile. Why? So the reason for this is, the reason will be that they usually have a trisomic condition. So they have a trisomic or a trisomy condition, trisomy of sex chromosome. That means such kind of males, or such kind of females rather, they have the presence of, and they have a presence of a kind of, you can say that such kind of females usually, or such kind of males usually have a kind of an overall masculine development that is called gynecomastia and is considered to be sterile, because the trisomic condition that has been observed over here is your you can say XXY condition. All right, so they have an XXY condition. That means or you can say 44 plus XXY condition, because this 44 is going to indicate the autosomes. This is autosome, and there is an additional X chromosome. An additional X chromosome, resulting into trisomic condition, which is 47. Clear? So that's why the such such kind of individuals are considered to be as sterile. All right. While Turner syndrome or Down syndrome, that is also a different kind of a condition. Down syndrome also is a trisomic condition, and this this is usually in case of those individuals where you can say, there is there is overall the overall brain development is very less and all those things. So each of the syndromes, they have different, like in case of Turner syndrome, there is exo condition. Then there is an trisomic condition, which is observed in case of a Down syndrome also. Then Edwards syndrome also. Different conditions are there. All right. But in case of a Klinefelter syndrome, if we are going to see, there is the overall masculine development that results into the body, and that's why such kind of individuals are sterile. All right. So that's why option A is the correct answer. Now, question number 79. Now, question number 79 says that which of the following is the most important cause for the animals and plants being driven to extinction? All right, that means you have to identify the main important cause that is responsible for the animals and plant loss, or not loss, we can say, but it is actually responsible for extinction. All right. So, there are basically four parameters. There are four important, four important rather parameters that is responsible for biodiversity loss. So loss of biodiversity is because of certain reasons, and in which one of the most important reason for the cause of extinction is the habitat loss and fragmentation. So there is habitat loss and fragmentation. Then there is alien species invasions. Alien species invasions. Then overexploitation. That means overexploitation of natural resources. So overexploitation of resources. And the fourth is co-extinction. So these are the main reasons for which the loss of biodiversity is being taking place, and the animals and plants are being driven to extinction. All right. So out of all these four statements, the very first important statement is the habitat loss and fragment. This is the prime cause. So you can see that out of all the options, option D is considered to be as the correct answer. That is driving, because habitat loss basically means that the natural habitat of the plants and animals is lost because of urbanization, or because of industrialization, or because of any other purpose that the humans are that the humans will use. So that's why it is actually driving all the animals to move towards the urban areas, or to move towards the city, and plants are actually getting extinct. So this is the one of the important main reasons. So option D is the correct answer. Now, question number 80. Now, question number 80 says that what triggers the activation of protoxin to active Bt toxin of Bacillus thuringiensis in the bollworm? All right. So now let me tell you that basically this is a question that has been a kind of, this is basically a question that requires an explanation, because Bacillus thuringiensis is basically used as basically used to control the bollworms in the cotton plants. And here they have a very unique ability, because these Bacillus thuringiensis, during their process of growth, they form a particular protein crystals in their body. All right. And these crystals, inside these crystals are found certain insecticidal proteins. And you will be very surprised to know that these insecticidal proteins doesn't harms the bacteria. That means doesn't harms the bacillus, but it is going to harm that particular ballworm, that worms, that will be ultimately ingesting those protein crystals. All right. So how the process goes on? So this basically goes like, you can say that Bacillus thuringiensis forms protein crystals around their life. So protein crystals during their growth phase. So during their growth phase, they are going to produce the protein crystals. Now these protein crystals contains toxic insecticidal proteins. So protein crystals contains insecticidal protein. That means those proteins which will be only and only harmful to that target insects. So contains insecticidal proteins which are present. These are actually toxic. All right. So insecticidal proteins which are present in bacteria as inactive protoxins. So these proteins, that means insecticidal proteins, these are found in the bacteria as inactive protoxins. All right, inactive protoxins. But the question that arises in our mind is that, so how these inactive protoxins will be converted into active toxins? So these will be only converted into active toxins once they are being ingested by the, bollworms. So these protoxins, once ingested by insect, gets converted into active. This is going to get converted into active form of toxins. Why they are converting into active form of toxins? It is because of the presence of the alkaline pH of the gut of the insects that is activating it. All right. So due to, remember, due to alkaline pH of the gut of insect, which actually solubilizes that protein crystals. And once these protein crystals, they are solubilized, the insecticidal proteins are released. So it is due to alkaline pH of the gut of insect, which solubilizes protein crystals. So this is going to solubilize the protein crystals. Now, once this toxin gets activated, once toxin is activated, this is going to bind to the gut of the insect. It binds to the surface of epithelial cells of midgut. This actually binds to the epithelial cells of the midgut and thereby creating pores in them, pores in the cells, and thereby creating pores that causes swelling and lysis of that particular cell. That means the cell is going to burst out and eventually death. Eventually, just a second. So eventually death of insect. All right. So this is how they show their effect. So it is because of the alkaline pH of the gut, this entire process takes place. So that's why the correct option is option number B over here, which is alkaline pH of the gut. All right. Now, the next is question number 81. Now, question number 81 says that you have to select the correct sequence for the transportation of sperm cells in the male reproductive system. Now, let me tell you that all these structures that has been given, it starts from the important structures. All these structures, we have to identify the entire male reproductive system. So how it is being transported. All right. So sequence. If you have to identify the correct sequence, all right. So the correct sequence is going to start with the very important cell, and that is the seminiferous tubules, because these are the only cells which are going to produce sperms. So it starts with seminiferous tubules, which are the main cells or main important site inside the testis producing sperms. So it is seminiferous tubules. Seminiferous tubules will be followed by retitestis. Then it will be vasa efferentia. Then there will be epididymis. Then vas deferens, which is also considered to be as sperm duct. And then there will be ejaculatory duct. Where the sperms will be stored along with the secretions from the accessory glands. All right. Now let me tell you that all these four, they are the category of accessory ducts. These are accessory ducts in sequence that will be carrying the sperms along the passage. Then there will be ejaculatory duct. Ejaculatory duct is further followed by urethra. And urethra will be further, leading to the final passage where it will be released, and that is called urethral meatus. All right. So this is the correct sequence. Now let us take a look. So it is going to start with seminiferous tubules, and then this is going to start with retitestis. So as you can see, retitestis, then vasa efferentia, epididymis, vas deferens, ejaculatory duct, urethra, and urethral meatus. So option A is the correct answer over here. All right. Now, next question is question number 82. Now, question number 82 says that persistent nucellus in the seed is known as. Now, this is a direct question that has been asked, all right? And this is a question from your sexual reproduction in flowering plants, basically. So we know that persistent nucellus is basically considered to be as perisperm, all right? So persistent nucleus is called perisperm. Remember, perisperm. And one of the best examples of perisperm includes, black pepper. Then there is beetroot. All these are the examples. So there is beet. Then there is black pepper, where the nucleus remains persistent even after the process of fertilization has taken place. Nucleus, that means the part of the your this one ovule. So black pepper. All right. So that's why the correct answer is perisperm over here. However, this tegmen is basically the hilum. Let me tell you about hilum. So hilum is basically the point of attachment. All right. In the seed. Then tegmen is basically the inner covering of the seed. Testa and tegmen, there are two coverings. So outer is called testa and the inner is called tegmen. Then chalaza is that point. It is basically the basal part of the ovule, chalaza. All right, that has the complete covering of integument. So again, this is not an option. These all three are not the option. So option A is the correct answer. Now, question number 83. Now, question number 83 says that which one of the following equipments is essentially required for growing microbes on a large scale for industrial production of enzymes. Now you have to grow, these microbes on a large scale for the production of enzymes. So for the production of enzyme, that is the one thing that it is required is basically the bioreactor. Again, this is also a very direct question that has been asked, and you might have studied it also in case of the chapter of your microbes in human welfare, that bioreactors are a kind of a large tank like structures where the microbes are grown on a very, very large scale for the production of industrially important or commercially important enzymes, you can say. All right, so basically we can say that bioreactors are large tanks meant for large scale production of microbes. Large scale production of microbes for industrial preparation of enzymes. Preparation of enzymes. All right. Now, here the other options that has been given are the sludge digester. Now, this sludge digester is basically used in sewage treatment. All right. So this is primarily used in, these are also large tanks, but that is primarily used in sewage treatment, not for growing the microbes, or that is meant for industrial production of enzymes. So again, this is not an option. Then industrial oven also is not an option, because these are not large. These are not used for large scale. So again, this is not an option. And BOD incubator, BOD basically is the biochemical oxygen demand incubator. And this is also used in case of pollution. This is actually used in case of a pollution indicator, or the level, or to check the level of pollution in the water bodies. All right, so again, this is not an option. So that's why the correct answer is supposed to be the option C over here. Now, question number 84. Now, question number 84 says that you have to match the following structures with that of the respective organ or locations. Okay, so you have to match the following structures with their respective locations in the organs. So some of the organ structures are given in case of the human body, and you have to identify the exact location. So let us just take a look. So the first option is crypts of Liberkin. So crypts of Liberkin is a part of small intestine. All right, that basically helps in increasing the surface area of absorption. So crypts of lubricant that is found in small intestine. Clear? This is found in case of small intestine. So this is going to relate with option A over here. Then there is Glison's capsule. Now Glison's capsule is basically found in case of liver. All right. So this is found in liver. Again, option B will be Glison's capsule. Glison's capsule is a kind of a sheath. It's a covering, let me tell you. That is a kind of a sheath or a covering that covers the liver. So that's why it's called Glison's capsule. So this is found in liver. Yeah. Then option C says islets of Langerhans. Now islets of Langerhans, this is a part of pancreas, which is option A. All right. So this is C. So islets of, because the basic unit, or you can say the important unit, the cells of the pancreas, that is called as the islets of Langerhans. And islets of Langerhans is further made of your alpha and beta cells. So islets of Langerhans. So this is basically the endocrine part of pancreas. Yeah. And the D option, Brunner's gland. So Brunner's gland, they are basically found in the submucosa layer of the duodenum, which is the small intestine part. So again, which helps in secretions only. So Brunner's gland. Brunner's gland. Are found in submucosa of duodenum. So this is the case. All right, so let us just match up with the different options. So A is going to match up with 3. That means third one. And then B is going to match up with 4th. That means this one. Then C is going to match up with 1, which is this one. And D is going to match up with 2, which is this one. So option B is the correct answer over here. All right, now let's move on to the next question, which is question number 85. So question number 85 says that what would be the heart rate of a person if the cardiac output is 5 liter, blood volume in the ventricles at the end of the diastole is 100 milliliter, and at the end of the ventricular systole is 50 milliliter. Now let us calculate this, numerical on the basis of the formula that we have already studied, and that is called cardiac output. Now, if you people remember, then cardiac output is equal to stroke volume. It is equal to stroke volume into heartbeat or heart rate, you can say. Heart rate. Heart rate. Clear? Now, the cardiac output, that given conditions that has been given over here, given cardiac output is 5 liter over here. All right. Or we can consider it, if it is 5 liters, so if we just convert it into 5 liter, that means it will be 5,000 milliliter, we can say. All right. Now the blood volume in the ventricles that has been given at the end of the diastole that you can see. So blood volume in ventricles at end of So the blood volume in the ventricles at the end of, diastole, if we consider it, diastole. So this has been given, let me see. So this has been given 100 ml. All right. So this has been given as 100 ml. Now blood volume at the end of, blood volume in the ventricles at the end of systole, if we will see to it, systole, then this has been given as ventricular systole. This has been given as 50 ml. This is 50 ml. So if we just need to find out the stroke volume, therefore you can say stroke volume will be 100 minus 50. That means the volume of blood pumped by each ventricle, each per beat. So that is called as stroke volume. All right. So this is going to be as 50 milliliter. So we have found out our stroke volume. So if we just calculate it in the formula, then 5000 ml is equal to 50 ml into heart rate. This is the formula. Clear? So therefore heart rate will be 5000 divided by 50. That means 100 beats per minute. All right, so this will be 100 beats per minute. That will be the calculation. All right, so let us take a look into the answer. So it will be option number B, which is 100 beats per minute. Clear? Let's move on to the next question, which is question number 86. Now, question number 86 says that xylem translocates. Okay, so xylem is one such conducting tissue, permanent tissue, complex permanent tissue, which is primarily responsible for not only translocating water and minerals, but it also translocates all the dissolved constituents in it. And all those dissolved constituents includes water also, minerals also, some organic nitrogen also, and some of the dissolved hormones also. So all right, so that's why the correct answer, very direct question that has been asked, so that's why the correct answer will be that xylem is responsible for translocating So this xylem is responsible for translocating not only water, but water, then mineral salts, then some organic nitrogen, and hormones. All right, so that means option C is the correct answer. Yeah, let's move on to the next question, which is question number 87. Now, question number 87 says that what is true for the golden rice? That means which of the following statement is true for the golden rice? All right, so it is asking about golden rice. So golden rice, it is pest resistant with a gene from Bacillus thuringiensis. It is drought tolerant, developed using agrobacterium vector. It has yellow grains because of a gene introduced from a primitive variety of rice, or it is vitamin A enriched with a gene from daffodil. Now, let me tell you that golden rice is, out of all the statements, the very popular statement that golden rice is enriched with. Now, golden rice is basically a transgenic product. All right. And it is a transgenic plant, or a transgenic plant product, you can say, which is highly enriched with vitamin A. That means this one, vitamin A enriched, and that too with a gene, it is having it from the daffodil, and it is also rich in the carotene as well. All right. So that's why we can say that golden rice is having a combination of, it is vitamin A enriched. So it is vitamin A enriched, and along with that, with a gene from daffodil, and along with that, they are also carotene rich. Carotene rich. All right. So that's why option D is the correct statement. Clear? Now, question number 88. Now, question number 88 says that purines which are found both in DNA and RNA. Now, let me tell you that, there are two categories. Any genetic material is basically made of two components. One is called purine and the other is called pyrimidines. So purine is a combination of adenine and guanine. All right. While pyrimidine is a combination of thymine or uracil and cytosine. Clear? So this thymine is found in DNA, and this uracil is found in case of RNA. That means this is having a combination of ATGC and this is having a combination of AUGC. Clear? That is the case. Like this. So let us take a look. That is found in both DNA and RNA. So it is adenine and guanine here. So you can see the very first option is correct. So that means this one. All right. Now, next question is question number 89. Now, question number 89 says that which of the following protocols did aim for reducing the emission of the chlorofluorocarbons into the atmosphere? All right. That means which of the following protocol is actually responsible for controlling the emission of the CFCs? Now, for control of the emissions, different kinds of protocols have been proposed. But for controlling CFCs, if we talk about, then Montreal Protocol is the protocol that has been that has been that came into effective, aiming for the purpose of reducing the emission of CFCs, because these CFCs, they are highly responsible for ozone depletion in the stratospheric zone. And this protocol has been signed in Canada. All right. So it is the Montreal Protocol. Montreal Protocol aimed for reducing emissions of CFCs. So emission of CFCs, which are chlorofluorocarbons, in atmosphere. All right. Now, what is the reason for this? Now, the reason for this is because these CFCs, since CFCs are responsible for ozone depletion in stratosphere, which is a protective covering that protects us from the harmful ultraviolet rays of the sun. The ozone depletion in stratosphere. That's why. Clear? And as I already told you that this protocol has been signed in Canada. Yeah. So Option D is the correct answer. Now, question number 90, which is the last question. Now, this question says that placentation in which the ovules develop on the inner wall of the ovary or in the peripheral, you can say part, all right, or in the periphery, you can say, or in the peripheral part. So, let me tell you now, this placentation process is one such process that indicates with the arrangement of the ovules inside the ovary. All right. Now, if it is talking about that the development of the ovule is in the inner wall of the ovary or in the peripheral part, that means in the periphery. So definitely it is the parietal placentation, because parietal placentation is the placentation in which the development of ovules takes place, in the inner wall of the ovary or in the peripheral part, you can say, just like in case of mustard or argemone. All right, so we can say that in parietal placentation, the ovules develop on inner wall of ovary. So these ovules, they usually develop on inner wall of ovary or peripheral particles, just like in case of mustard and argemone. Example includes argemone maxicum. Argemone maxicum. All right. So this is the case. So that's why the correct answer will be parietal placentation. Free central will be in the free central, as the word is suggesting. This will be in the center. So definitely this is not the correct option. Basal means the basal placentation at the base. So definitely this is also not found in the periphery. And axile means that in case of the axile placentation, the arrangement again is on the central portion. So that's why this is also not the correct option. So the correct answer is option number B over here. All right. So with this, we have completed the 90 questions of your NEET 2019 paper.